Hello, my name's Christopher Bravery. I'm a consulting regulatory scientist at Advbiols. Due to the limitations of only 20 minutes for this talk I gave at PEGS on the 11th of November, I decided I would like to provide an extended version where I have an opportunity to explain a few more of the aspects that I had to gloss over a little bit in the shorter version. So I hope you'll enjoy this. So while this isn't a generalized talk on potency assays, it's always important to start by just gently reminding ourselves what a potency assay is. It's a measure of relevant biological function or functions indeed, because we would expect more than one function from many of the advanced therapies. And I should emphasize here that I think there's sometimes a misunderstanding or a sort of assumption that other biological products are simpler. Actually, monoclonal antibodies commonly need more than one potency assay because they do work through more than one mechanism of action: antibody-dependent cytotoxicity, complement-mediated cytotoxicity blocking, etc. So it isn't unusual to have more than one potency assay, or perhaps not as unusual as maybe some mistakenly think. It's also really important to emphasize that potency is not efficacy. Indeed, if you think about it, potency is a measure of the activity of the active substance. And that activity will act upon the body to induce the efficacy. So the relationship between potency and efficacy is by no means necessarily straightforward. Because some of the things that I read make me wonder if people fully understand that principle. And then I think it's also really important to call out the elephant in the room when it comes to measuring potency. This problem shouldn't exist for vector-based advanced therapies. So that's ex vivo gene transduced. That's vectors as drug products, or indeed vectors as starting materials. In both those situations, there's no barrier to creating a reference material for the vector. The only barrier is the cost of the batch of material that you lay down. And that's not a regulatory excuse, unfortunately. But when we look at cell-based medicinal products, be they genetically modified or not, viable cells just really aren't suited to being reference materials, because one of the fundamental properties of reference materials is they need to be stable. So while the cells may be stable when they're frozen, of course, they're very unstable when they're thawed. And they just have a habit of changing their characteristics depending on how they're handled. Having said that, one of the issues that I note is that all cell-based product developers seem to just dismiss reference materials despite on this basis. And really we need to find a solution for each specific situation. Unfortunately, we can't, it's not likely we're going to find any sort of universal solution. There is a publication that I list at the end where I ruminate on this in a paper that I wrote a number of years back, which sadly hasn't apparently had too much notice by people. But the absence of a reference material is really a significant metrological limitation to what we're trying to achieve when we develop a potency assay. So to help those of you that might be a bit puzzled by what I say, I mean, the first thing to say is that your reference material, and it has to be a batch of your product, there isn't, well, it doesn't necessarily have to be a batch of your product, but in general terms for biologics, it's a batch of your product because it is the thing that's the closest to what you're trying to measure. So you assign a batch some arbitrary units of potency, and then you retain that batch and use it as a reference material, as a calibrator, as another term for the same thing, to ensure that you can look at the relative potency from batch to batch. And this is important because potency, the units of measurement for potency are units. They are arbitrary units. We can't define them by normal physicochemical standard, international standard units like mass or length or time, etc. We have to define them in these arbitrary units. So the reference material becomes the units of your potency. And so you can see why, I hope, by that explanation, this causes us a problem for potency assays for cell-based products. So to try and illustrate that point with a simple example, until very recently, I mean the last couple of years actually, work had been ongoing for a number of years to replace this prototype kilogram. But the prototype kilogram was created in 1889, and it's made 90% platinum, 10% iridium, because they're very, very stable metals and they don't corrode. And actually, platinum has a nice property that it's quite soft. So even if you nudge it or bang it, you move the molecules around rather than knock them off. Because obviously, if you define this as a kilogram, which we did in 1889, every single measurement of mass since 1889 has referenced this. Even the US imperial measurements are actually defined in law based on the kilogram. So even if you do use pounds and ounces, they relate back to the prototype kilogram, or now the replacement of that. So this physical object is used when measuring the mass of a neutrino or the mass of the universe. And obviously, if its mass was changing, then this would be causing us all sorts of problems, particularly at those extremes. It doesn't have much effect when you go and buy a kilogram of bananas. And indeed, when they brought the secondary copies of the reference material back to the small building outside of Paris and compared, they were finding that the masses were deviating. Tiny amounts, I assume, but doesn't take much to affect big or small mass calculations. So this was just not really very acceptable, because if it had been lost, we would no longer have a kilogram. We'd have to recreate it, and that would cause all sorts of problems. So leaving that aside, in principle, your reference material for your product is the same, but we do use it for other things as well, and that can lead to a bit of confusion. So let's look at a typical scheme. I mean, I drew the paper that I wrote on reference materials, I drew something a little bit similar to this, but this is from Bioprocess International. So just to show to you, this is normal for biotech, and it is normal for all biological products if you're not familiar with it. So as you can see, right in the development phase, you should be laying down your first reference material. Indeed, most biotech developers would lay down some sort of reference material, even in preclinical. And for certain, you want to lay down one for first in man, and it should be a batch that was used for first in man studies. Because one of the other uses of the reference material really is as a reference to the quality of your actual product. And that, so that can be used for comparative purposes, and you can have a look and see what the quality was. And then as you go through development, when you eventually get to your pivotal studies where you're demonstrating safety and efficacy, you would also then lay down a brand new one for the commercial phase. So this is the phase 3 pivotal area here. And this is when you would lay down your commercial phase reference material. However, you will have assigned units of potency to the earliest one. So this one would be calibrated against the other one for the potency assay. But this then represents the quality of the material that demonstrated safety and efficacy in pivotal clinical studies. And that means that in the future, for things like comparability exercises, you can compare. So you don't want to waste the primary reference material, sometimes called the master reference material. You want to create working reference materials as well. So that's yet another batch of material or part of a batch that's laid aside, and that's used for the routine use. So it assumes that you can store the reference material for a very long time. Ideally, you'd want to be able to store that primary reference material for the whole product life cycle, which might be possible for some biologics. I don't know really how often the primary ends up being changed, but eventually it might run out depending on how much you were able to store. But the working ones you would qualify from time to time. Anyway, so the point is that we have this gap for cell-based products that we're not laying down some sort of reference material, and we really need to rack our brains for that because it's pretty important. This is a figure which is primarily based on data from Barkholt et al. I've given you the DOI at the bottom. Barkholt and et al. Are some of the CAT members from the European Medicines Agency, and they reviewed data from 19 submissions to the EMA and scored where the major objections come from, the types of issues. And as you can see, potency assays come out as something like the 5th most common problem, affecting up to 50% of applications. I think we could say on average around 50 or so, a bit more than 50% of applications. So they are problematic. So let's not deny that this is a challenging topic. So for the benefit of those that may or may not really have very much familiarity with potency assays, here's how we would love it to be for advanced therapies. I mean, in principle, for gene therapy, you might be able to achieve something like this as well. But certainly for cell-based products, we would struggle to come up with such nice, clean data. So this is GCSF, which it's INN, its international non-proprietary name is filgrastim. And there are actually a lot of filgrastims on the market, quite a lot of biosimilars now, which is one of the reasons why there is actually an international reference standard for potency created for the WHO. And this allows us to calibrate the potency and report the units of potency of this product, of every GCSF product, to the same units, which is obviously has a safety implication really for the clinical work when you're dealing with biosimilars, etc. So, but for most of us, most developers, the product they make will be unique on the market and nobody will be copying it. And by copy, I mean it has to be exactly the same. So you can show that 2 GCSFs have the same amino acid sequence. So you can conceptually accept that they're the same active substance, except that we, in a rather rigorous way with biologics, acknowledge that the manufacturing process alters the quality of the product. So it's not about quality now, it's just about what the active substance is, if you like, although the quality of the active substance is in there as well. But when you're looking at CAR-Ts, it would, for example, I think it would be a lot harder to argue, just because they're all directed against CD19, that they are, from a molecular point of view, the same product, because often we've got different signaling pathways and so on. But it might be, I suppose, theoretically possible that we could come up with some sort of reference material that was broadly suited to killing assays or something for CAR-Ts. But again, I think this is a complicated area, and it sounds easier than it's likely to be in practice. Anyway, the point of this slide is simply to show that when you alter the dose, keep increasing the amount of GCSF in the assay, you get an increased response. And we typically see this sort of sigmoid dose response curve. And then if, funnily enough, I couldn't really find any human data, which was a bit strange, or not anything that was as clear as this. So I apologize for this, but this is now neutrophil counts, which is your biomarker, if you like, for the efficacy of this product. And this is in primates, and they've actually got wild type GCSF in pink. And then the light blue, cyan, whatever color you like to call it, is 10 micrograms per kilogram per day. And the brown triangles below are 5 micrograms per kilogram per day. So you're getting something like half the response. So isn't that lovely? If only we could see something like that for our potency assays. Anyway, leaving that aside, so part of the reason I decided to focus on immunotherapies, measuring T-cell responses, if you like, so primarily CAR-T type potency assays, is shown in this publication. It just shows a number of different products and the type of assay they're using. And as you can see, cytotoxicity assays and interferon gamma release are two very commonly used assays. And I want to unpick some of the metrological problems with those two types of assay. So I had to find some sort of data to try and enable my sort of argument, my thesis, if you like, about why there's some issues with these products. And of course, inevitably, you can't find exactly the sort of data you'd love to find, but I did find this paper by Hagen et al. And this paper was just comparing 3 different methods for looking at interferon gamma. And that's by ELISA type methodology, looking at the release into the culture media, by ELISpot, which for those of you not familiar with what ELISpot is, it's basically a nitrocellulose membrane. The cells are laying on the membrane's covered in a capture antibody, and as the cell releases the interferon gamma, it's captured around it, and then it's sort of developed a bit like an ELISA, so you put an insoluble color, so you see a dot. And then flow cytometry, so intracellular flow cytometry. So they add a chemical, the name of which I can't remember, that traps the interferon gamma inside the cell over the duration of the bioassay. And then the cell is fixed and permeabilized and stained for interferon gamma. So you can see how much interferon gamma had accumulated inside the individual cells by flow cytometry. So this is a comparison of 3 apparently orthogonal methods for looking at interferon gamma. And that's an important point in itself that you should be doing with your potency assays. You should be looking for orthogonal ways of looking at the same form of activity in order really to corroborate that what you're measuring is reliable and meaningful, and there isn't some sort of artifact of the assay itself. So in this study, they don't use purified T cells. They've actually just isolated peripheral blood mononuclear cells. But what's nice about it is they've used 5 different healthy donors. And then they've, rather than use a specific antigen to stimulate them, they've used polyclonal activation using via PMA and ionomycin, which for the T cells mimics signal 1 and signal 2, so it should drive them to proliferate. And for non-T cells, well, probably it will do something to them too, because these are 2 non-specific chemical signaling molecules, artificial signaling molecules. I don't really know how to call them, mitogens maybe. Anyway, so let's look at the ELISA results first, because in my experience, this seems to be the the most common approach for potency for immunotherapies, perhaps. But also, I chose this partly because measuring cytokines is also undertaken for a number of other products. And I think some of the general principles that I'm picking away at here will apply for other sorts of use of cytokine release type assays. And so I do think you need to think a little bit carefully about them. But anyway, in principle, to start with, as you can see, as expected, you see quite a significant variation in the amount of interferon gamma that's released from only 48 picograms per mil up to 1,449. And this is in itself a problem because one, what are we measuring? Well, we're ending up reporting in picograms per mil, which are SI units. So this isn't a potency unit of measurement. And we have no reference material to compare it to, so we can't give a relative potency. And so this in itself makes the result much more difficult to deal with, because in essence, what we should be doing is saying we allow 100% plus or minus 10 or 20% of something of potency. But here we can't really do it in that way. How would we report it in such a fashion? It's not possible. So we're tending to report in SI units and then setting a threshold. So this isn't really the ideal way to report potency, or it's not really reporting potency at all, to be fair. Anyway, so leaving that aside, so we all expected that because is inevitable. So let's move on to the ELISpot. So you can see from the number of spots that really you're getting the same sort of pattern, that the donors are highly variable, which is, again, what we would predict, sadly, for autologous products. But I think I wanted to point out also that the spots vary in size. And that means that they're varying in the amount of interferon gamma that any individual cell or cluster of cells is releasing, which, when you think back to the ELISA now, that will affect how much you measure in the supernatant. So when you think about T cells, even if this was 100% T cells, then there are T cell subsets, as simple as CD4 and CD8, which the ratio of which varies quite a lot normally amongst people, from, I think, maybe even as low as 0.5 to 1 up to 2 to 1 of CD4 to CD8. I mean, the ratios are quite variable. But there's also further subtypes of T cells which we know secrete various different types of cytokine. In fact, we define some of those by the cytokines they release, and not all of them will release interferon gamma, and quite clearly some release a lot and some don't. So it's the heterogeneity of our active substance which actually is making using something like cytokine release really quite problematic. If we could produce a very consistent product that always had the same distribution proportions of the various subtypes, we might have a better chance of finding that interferon gamma release was useful. However, as a slight aside before I move on to the next aspect, I found a different paper, and in fact I spotted this in a couple of papers on ELISpots. This is just to show that when you look at the distribution of the spots, they are normally distributed, which is sort of reassuring. I'm not sure it adds much to the argument, but from the point of view of counting spots, from the metrology of doing an ELISpot, it's useful to know they're normally distributed. Which then pushes me to perhaps point you in the direction of USP 1032. And when you're developing potency assays, do think about some of the other aspects that might affect reproducibility. I think the USB chapters on potency, etc., probably overemphasize the statistics, and particularly if you're applying them to a cell-based potency assay, where probably the level of statistics is beyond the reality of the data. But But it makes a very good point about looking at the distribution of signals across a plate to make sure that the way you're plating the product out and the plates you're using are giving you consistent results, i.e. you're not getting obvious effects. And any of you that are familiar with doing bioassays are probably quite familiar with deliberately avoiding the edge wells because of the well-known edge effect. Something you don't tend to worry so much about with ELISAs. In fact, most people do use the edge wells for ELISAs, but with bioassays, we tend to just put some sort of media in the outside or even just PBS to make sure that we're not getting any sort of funny drying effects near the edge. So it is important to look at that. And indeed, I found this figure in yet another paper, which actually made me smile a little bit, but I think is a very good piece of work to do. And this was basically for the plate reader for ELISpot and making sure that the plate reader was consistent. And so this figure represents reading the same plate 80 times over 7 hours to make sure that you're getting a nice crisp signal. And assuming those are tiny little error bars hiding in there, I think that the detection system was pretty effective. Anyway, so now let's return to our 3 orthogonal methods for looking at interferon gamma. And here we are now with the flow cytometry results. So this is now looking at intracellular interferon gamma. And as I mentioned to you, this particular study is on PBMCs. And PBMCs are typically maybe about 70% T cells. And those T cells will have all the subtypes that I was referring to. But if you look at the third column from the left, you'll see here that they've also included CD3 in the flow cytometry. So the negative cells have, up to 20% of the negative cells are also releasing some sort of interferon gamma. At least it's detectable. The flow plots, I think, also demonstrate to you in the same way as the dots did on the ELISpot that there's quite a big spread in the fluorescent intensity of the interferon gamma amongst the population that's positive. I mean, it's a spread of a couple of logs. So that could be really quite significant differences in the amount of interferon gamma produced by different cells. Again, not really helping us to feel confident that this sort of approach is going to give us reliable data. So now let's look at the correlation. I mean, the obvious correlation, if you were looking at my ordering of high to low that I included on the previous slides, the obvious correlation is ELISA versus cytometry, sorry, ELISpot versus cytometry, where in principle they are measuring the same thing. One is sort of trapping the interferon gamma inside the cells and looking at it after permeabilization. The other is allowing it to come out and stick to antibodies on the membrane around the cell. So we would have expected to get a good correlation here. And so if you were going to use either of these 2 methods and you were using the other one as an orthogonal method to confirm that you're getting a reliable measurement, then while a correlation of 0.7 is perhaps not ideal, I have no idea how well optimized these assays were, etc. Or indeed, it may be partly because this is partly because of these sort of impurities and so on. Who knows? I mean, there's all sorts of reasons why the correlation might not be quite as strong as we would like, but I think that's perfectly reasonable given these biological systems to argue that, yeah, okay, this sort of supports that these 2 methods are measuring the same thing. Different thing if you wanted to swap from one method to the other, of course, because actually demonstrating a strong enough correlation that you could consider the data together is another matter. I think you would still have to consider it a different method and adapt specifications, etc., if you were swapping between these methods. But, and here's the point that I was hoping to get to, there's really just no correlation between either flow cytometry or ELISpot with the ELISA results. So measuring secreted cytokine somehow doesn't correlate at all with the number of cells that are positive for the cytokine. And I hope I've sort of explained all the obvious reasons why I think this is true. And I think this is fundamentally the problem with measuring cytokine release from any product, unless you've got a very homogeneous population of cells, or that heterogeneity is consistent heterogeneity, that all the proportion, all the sort of subtypes of the cell are present in the similar proportions, then I think cytokine measurements are always going to cause you problems. But I'm not saying that you shouldn't try, because in some cases they may be perfectly suited. But I think fundamentally, from a metrological point of view, if you think about what you're measuring, then it's really no surprise, I think, that an ELISA for interferon gamma isn't very reliable. So now I'm going to move on to a different data set. And again, I must emphasize, I didn't even really read what the purpose of this paper was. I just found a paper with some useful data in that I could unpick. And it'd be pretty useful actually for picking at some of the issues that I see with these sorts of cytotoxicity assays in my work. So I think this was a completely research paper. So I don't think these are GMP batches or anything. But you know, in keeping with products on the market, products that are being developed, the transduction efficiency for these CAR Ts varies from about 26 to 42% transduced cells. So going back to my, concern about, purity of the product and how this makes actually measuring potency harder, now we have a problem where we've got high 90% of CD3s. So we probably don't have to worry too much about the non-CD3s in here. Hopefully whatever signal they generate will be minimal. But obviously you want to look. But we now have the issue that the cells we're interested in are less than 50% perhaps of the population, which means that if the non-CAR cells, so the untransduced T cells, are in some way stimulated by the bioassay, then they are going to contribute to the signal, and erroneously perhaps. So it's about specificity of the bioassay for the active substance, I suppose, is what I'm talking about. And that should be something that you look at in qualification and later confirm in validation. So that is an issue. So how do we deal with that? Well, I was interested when I looked at the Yescarta BLA documents. There was a specification that was highly redacted in one of the documents, but the units for potency were provided, and they were picograms per transduced cell, which means that it sounds like they've attempted to normalize for the transduction efficiency by dividing by the number of transduced cells. So I'll come back in a couple of slides to whether that looks like it would work or not with the particular data that I picked out of a paper. But first of all, let's look at a killing assay. So I actually started life as an immunologist as well, and I've done killing assays and so on, and I've used effector target cell ratios, both for MLRs and killing assays, stimulator responders, etc. So I'm familiar with this approach, but actually when I look at it from a pharmaceutical perspective, I find it puzzling when I see it in an IMPD, because sometimes I see that they're measuring potency at, say, 3 different effector target cell ratios. But that's just making it a little bit harder to actually report the potency, because we can set 3 different specs at different effector target cell ratios. It tends to get a little bit confusing. But actually, when I looked at the experimental details of this particular experiment, the targets are consistent in all wells. There's the same number of targets. It's only the effectors that are altered, which means it's a dose response curve, which is really what you want to show. But it's sort of slightly obscured by these effector target ratios. But that means that in the way that this particular figure is presented, it's not been put on an XY scale, really. I think they've used the wrong the wrong type of graph in Excel. These are just evenly spaced as if they were going to be bar charts or something. But actually, in effect, it's a logarithmic scale, which means that they shouldn't really have this 0 here, because you can't put 0 on a logarithmic scale. And to draw a straight line up is also rather a bad assumption, because we wouldn't necessarily expect a straight line. I mean, as it's logarithmic, we would expect this to be a straight line, but at some point we would lose that linearity. The other thing that was a bit odd about this particular graph is the untransduced cells also give really quite significant killing. There were 2 other, I think, figures in the paper where the untransduced cells didn't produce anything like that amount of killing. So this may be a peculiarity, but it was another good reason to pick it as a figure, because, you know, there are various reasons why this might be the case. One possibility is these untransduced T cells are alloreactive for the target cells, or the target tumor cells, which will be allogeneic to the product. And my textbook in immunology tells me that something between 1 in 10 and 1 in 100 T cells in the peripheral blood are alloreactive. So we could expect an alloreactive effect from the untransduced population. It seems in this situation, maybe it was really pretty extreme compared to the other figures that's shown. But fundamentally, this needs, the real problem with this figure is it just needs to be redrawn. And so I did that as best I could by reading the dots off of the graph. So you know, this is a pretty rough redrawing. Please don't think that I have the data or anything. And so now I've just changed it. I just calculated the number of effector cells on the x-axis, and I've left survival for the time being on the right, on the y-axis. But of course, survival for a killing assay is, in my view, a rather odd way of looking at it. Normally, we would put killing on the killing for a cytotoxicity assay. So maybe the authors were thinking of survival of their patients when they decided to plot this out. But actually, we want to see death of the tumor cells. So it seems strange that they didn't choose the word killing and flip the figure around. But one of the problems that immediately comes to light when you put it in this way is that we're not seeing the full dose response by any stretch of the imagination. Actually, for the untransduced in orange, you can see that we can see what the EC50 is, the 50% killing rate. And we know, therefore, the number of effector cells that will kill 50% of those target cells. But for the CAR T cells themselves, we're nowhere near the EC50. We're well beyond it. Even the lowest concentration of cells they added kills more than 80% of the target cells. So from a metrological perspective, this is an insensitive assay, because you could have big changes in the effector cell number, or the number of effector cells that were effective. And it would have almost no effect in this area of the graph on the amount of killing. And we really would like our potency assay to be quite sensitive. In particular, if you're looking at stability, you want to be able to detect stability. So my guess is that this wouldn't be very stability indicating used in this way. If you could move the graph down to the more sensitive point, so it looks somewhat more like the orange one, but preferably going from much higher to much lower, seeing the full range, then you would know in that range of the graph, a kind of a small change will lead to a bigger change in signal. And as I say, fundamentally, really, it should be plotted like that, as percentage of killing. And again, this makes it perhaps even more obvious then that really, even down to even if we went a bit further, I think we'd find that, well, maybe I'll just put that graph. So this is what the graph I think should look like. And it doesn't leave much space, which does make me wonder if there was something a bit odd about this particular set of data. There was an awful lot of background killing for some reason or another, non-specific, and that wasn't allowed for. I don't know, because I don't have the raw data. So, you know, maybe I pushed this a little bit further than I could. But anyway, if we put it onto a log, we should be able to draw a straight line, although eventually it will bend. And as you can see, I think if we got down to one cell, we would still have more than 50% killing. So I think there's a problem there. But the real message for this is just that they're not, if they were going to use this as a potency assay, I don't want to preempt whatever the purpose of the paper was, then they're not using it in the sensitive part of the range. Now, interestingly, they also looked at interferon gamma release from, I think, the same wells of the same experiment. So they just collected some of the supernatant and also did an ELISA type methodology. So again, they're presenting it as effector target cell ratios, which is not helpful, but we know now that we could replot it like that with the effector cell number along the bottom and the response. So we've now got a dose response. And we are seeing a bit of a curve, and we are getting a good dose response, which is now then a good time to come back to my comment about Yescarta dividing by the number of transduced cells. So if I take this point here, roughly in the middle of the figure, and use the amount of picograms per cell at that point, and then plot it forward and backwards, you basically would see that if it was a linear relationship across that range. Now, I wouldn't expect it to be a linear relationship across the entire range. And indeed, I've already made the point that they're far too much to the right here, and they needed to go further to the left in order to see the dose response, the full dose response curve. And you can see from the bit I've projected to the left side that maybe it's nearly linear for a portion of time. And so if you were within that linear region, then it might be reasonable to divide, and perhaps that's what what the developers of Yescarta were able to do. But as you can see, it's not necessarily going to expect it to be a linear relationship. And again, I think from theoretical arguments, you can say that it might not be a linear relationship. So that's it, really. They were my thoughts for you to dwell on and think about, with a little bit of extended thinking from the presentation I gave on the day. The elephant in the room is, of course, that we don't have a reference material for generally for cell-based products. I know of one company, I think, that said to me that they were developing one, but I have no idea how that went. It's not a company I was consulting for. And largely I find that people are not really sort of giving me much sense that they really understand that it's a massive limitation not to have one. I have seen a situation where, at least once, I think, where a company has chosen to use some of the previous batch to compare to the next batch for an autologous. And that sounds okay until you realize that actually over a period of batches, batch after batch after batch, I think you could still very easily get drift, because A equals B, B equals C, C equals D, D equals E, but does A still equal E? It's the same sort of issue we have with comparability. When we make multiple changes over long periods of time, we can get process drift. And here my worry is analytical drift. However, that's another problem and not really the focus of this talk. But hopefully I've showed you that the value of looking at orthogonal methods for understanding where there might be limitations in what you're doing. I haven't really given you an answer to the problem, but I have hinted to you that I don't think cytokine release methods are particularly brilliant and are likely to be problematic, but I shouldn't necessarily deter you from trying them. But I would think about them very deeply before you bother, because I think you might find that they're not very practical in real life. if you're going, I think probably cytotoxicity should be more reliable, with some reservations, because I've seen slightly less cytotoxicity data. But it's really important for any of these assays that you do a full dose response, so you can see the maximum dose, the minimum dose. You can find out where the EC50 is. And when you're comparing different results from different batches, at the very least, you should be looking for some sort of symmetry around the EC50, and that they're looking parallel. And if you're getting funny shapes to those dose response curves, then it's hinting that there's something not quite right about the methodology. It's not being consistent. Anyway, I hope that was helpful, those extended thoughts. And I just emphasize finally, once again, that please don't take any criticism if you're the authors of any of the papers I've cited. I've just used your data to try and illustrate my own thinking. And indeed, I didn't, in most cases, pay much attention to what the actual purpose of the paper was. Apologies for that too, I suppose, because it sounds rather dismissive, but I wasn't looking for the papers for their content as per se, just for some useful data to play with. Thank you very much.